How's it going, everyone? This is MedCat here, and today we're going to be talking about histone acetylation and DNA methylation. Two different ways we can regulate the DNA within side of our cells. First things first, we're going to take a look at acetylation. This is a bit more high yield, so I want to cover it first and a bit more in depth. Let's take a look at this side chain here that's coming off of our histone octamer. Can you recognize this side chain? You should be able to, because amino acids are very important to know for the MCAT. This is going to be a lysine residue, whose three-letter abbreviation will be LYS, and one-letter abbreviation will be K. This is a basic amino acid, and if none of this is making sense to you, amino acids are very foreign, feel free to click in the upper right-hand corner to my link on the amino acid playlist, because that is the #1 highest yield content area on MCAT. Definitely need to know that. But assuming you do know that this is lysine, we know that lysine, being a basic amino acid and having that high pKr for its side chain, will be positively charged. Histones are very basic and very positively charged, therefore. And the thing that's nice about that for a histone is it allows a very simple positive and negative interaction between the histone and the DNA. For, after all, histones are DNA binding proteins, a type of nucleoprotein. And the reason that DNA is negatively charged is going to be because of those phosphodiester bonds and those oxygens that are going to be negatively charged in that sugar phosphate backbone. Okay. So just really, at the end of the day, it's as simple as a positive and negative attracting each other, which is what allows histones and DNA to come together. However, if we add an enzyme, perhaps to solution, called histone acetylase, which will be found in the nucleus, this will acetylate that lysine residue, aka amino acid, so that it will neutralize that positive charge. Now we've added this acetyl group, and by the way, this is an acetyl group, sometimes abbreviated, especially in organic chemistry, as AC. This acetyl group has neutralized that positive charge so that there is no longer that basic positive-negative Coulombic interaction there. Therefore, the DNA is going to be more free. It's going to be less tightly bound, therefore what we call more euchromatic, or more open to transcription factors and RNA polymerase. Therefore, we can express more of the genes in that DNA. Versus when that Coulombic interaction is here, that is going to be more heterochromatic and less open to transcription. Therefore, if we want less transcription, we can add something called a histone deacetylase, an enzyme that, of course, deacetylates that lysine residue to make positively charged lysine again. And by the way, this can also happen for another amino acid called arginine, although when we're learning it, lysine is usually the focus because it's a bit more common to occur in lysine. However, it can also occur in arginine, another one of our basic amino acids. Now let's move on to DNA methylation. As a general rule, DNA methylation is going to generally silence genes and make it harder for that transcriptional machinery, namely transcription factors and RNA polymerases, to actually access the DNA. And how it actually does this is we'll add a DNA methylase, and it will usually silence two different nitrogenous bases. Now hopefully you should be able to take a look at these and tell what they are right away. But if you don't, again, feel free to click in the upper right-hand corner to my video on memorizing your nitrogenous bases, which is an important part of the MCAT, but often an overlooked part of studying for the MCAT. So you should be able to recognize this as adenine and this as cytosine. And we're going to methylate these atom positions specifically right here. And it might not be super high yield to know that these specific positions are methylated exactly at these positions, but do know that it's going to be adenine and cytosine predominantly that will be methylated. And remember that for every adenine, there's going to be a thymine, and for every cytosine, there will be a guanine here. One last thing I want to mention is that even though DNA methylation generally silences genes, we also want to know that upon occasion, enhances transcription. So depending on where it is in the genome, methylation could actually enhance that transcription a little bit. Finally, let's take a look at a double AAMC-derived practice problem that I've written based on a real double AAMC problem. And I'll give you a chance to go through it, so feel free to pause the video and try it yourself before we go through it. So assuming you've had a chance to go through it yourself, the answer here is going to be B. And the reason for this is if we draw out our histone, let's say with lysine that has not been acetylated, so 1, 2, 3, 4 carbons, and our amine, which is protonated here with a positive charge. And then we go through and we acetylate that. Give me a moment to copy and paste this here. If we acetylate this, this is what we'll be looking at now. We've gotten rid of a negative charge. Therefore, our isoelectric point, or pI, which again we should know as kind of our pH, where our molecule of interest, in this case a protein, is predominantly neutral, or the greatest concentration of our protein will be neutral. To get to that pI, we're going to have to move up in pH less, because to neutralize this positive charge, we would need to go past the pKr of lysine, which is going to be pretty high at around 10, being a basic amino acid. Whereas if we neutralize this, we don't have to keep going up past that pH there. Okay. And of course, there are going to be a lot of other side chains that we have to deal with, but if we neutralize a lot of these lysine residues, as well as maybe some arginine residues, we're not going to have to go as high in the pH to get that protein to be neutral. Therefore, our pI would actually decrease. If that doesn't make sense, we can also think about it the opposite way. If we go back, if we start with an acetylated lysine and then add more positive charge, we're going to have to increase that pH in order to get that molecule to be neutral. So generally, when you add basic amino acids, you are going to increase that isoelectric point. And when you add acidic amino acids, you are going to decrease that isoelectric point. That's it for today's MedCat video. Feel free to hit the like button, subscribe, and check out my comprehensive amino acid playlist, which can be found in the link in the description below.